Hello everyone, my name is Sishuo. I'm from uh, Chinese University of Hong Kong. I'm very happy to have this opportunity to share with you some recent progress uh, on molecular clock dating using mixed substitution models. Um, so let me start from this introductory slide uh, about the some basics of Bayesian relaxed molecular clock dating. On the right-hand side of the equation shows the three key elements of Bayesian molecular clock dating, which is the time prior, uh, indicated by f of t, rate prior, f of r, and the last thing, likelihood function, which is to calculate the phylogenetic likelihood. Uh, while there have been quite a number of studies that have been done in terms of the first two things, particularly the time prior, which is about how to calibrate the time tree based on the fossil records. It seems to me that the last thing, the phylogenetic likelihood, has received relatively less attention in molecular clock dating. And in calculating the phylogenetic likelihood, we know that substitution matrix plays an important role. The reason is because usually, if we use different substitution matrices, we may get different likelihoods of the data and also uh, a different estimate of the parameters, the topologies and branches of the tree. Um, um, Importantly, for deep time phylogenies, we know that evolutionary pattern becomes more complex. So even within the same gene, the different sites can evolve in different patterns. For example, some they evolve in they may favor the JTT model, whereas others favor the WAG model. So there's a very good way to specify this and account for this uncertainty, which is to assign them different ways. And this is considered as the mixture model. And so specifically speaking, in the following slides of my presentation, I would like to focus on the so-called profile mixture model. So what is the profile mixture model? So it is the substitution model where we have more than one categories of the uh, equilibrium frequencies indicated by uh, calligraphic letter I. So if we have more than one category of the equilibrium frequencies over the alignments, we are specifying a profile mixture substitute model, which is made famous by the fellow-based CAT model and the C60 model available in IQ3. And so why is this important, especially for deep time evolution? Um, it's because first, it fit empirical data better than the non-mixture homogeneous counterparts, and second, it generally speaking make more biological sense, and last, there's no need to know uh, in advance in terms of how to partition the alignment, how to divide your alignment in different subregions. Let's have a look at this simple example. As pointed out by this arrow, we see that this column, this site, seems to have only two amino acids that are negatively charged. So in the eyes of a non-mixture model, it will interpret this as a result of short phylogenetic distance. While in contrast, in the views of a mixture model, um, this site doesn't mean too much, because it's very possible that it evolves over a very long evolutionary time because only negatively charged amino acid D or E are allowable at this site. So even if you see two identical sequences at this site, it doesn't mean too much about the phylogenetic distance. As a result, we often find that using a mixture model, we get a longer branch length estimate. And for this empirical example, the authors, Moody et al., analyzed the length of the branch that separate the bacterial domain and the archaea domain, which is probably the deepest branch in the tree of life. They found that using a mixture model, it much better fits the data, and also the branch lengths are much longer than estimated under the homogeneous non-mixture model. So here we know that using a mixture model for deep time phylogenies, we get much longer estimates for the branch length. And branch length is the product of time and the average rate of the branch. So how would the increased branch length estimate affect uh, the time and the rate estimate? So this is the question we are going to answer. Uh, Directly calculating the phylogenetic likelihood under complex substitution models is very challenging. However, very fortunately, we have the very fantastic approximate likelihood approach implemented in MCMC tree. So briefly speaking, so this method approximates the log likelihood of the phylogenetic data by using the tension. Uh, and we know that at the maximum likelihood estimate, the gradient is equal to zero. So briefly speaking, roughly speaking, we can ignore the gradient, and now our focus is how to estimate the hazing evaluated at the maximum likelihood estimate of the branches theta hat. Unfortunately, the panel doesn't implement mixture model. So the key is if we can find a way to estimate hazing under the complex mixture models, we can further import it into MCMC tree using the approximate likelihood method for dating. Um, so recently, there have been two studies independently done by different groups of people. One is from the IQ tree group. The name of the toy is called IQ2MC. The other from me, collaborating with Andrew Mead, and it's called Phylo hazing. So the idea is to calculate the hazing matrix evaluated at the maximum likelihood estimate of the branch length, theta hat, by using the numerical method called finite difference method. Okay, so actually what is happening in these two software is slightly more complex compared to what is shown here, but the idea and the logic is very similar. Um, so in the following slides, I will focus on my own study, but I'll go back to the other exciting study at the end. So our question is, how does profile mixture model, namely LG plus C60 plus G? So here we specify 60, we assume 60 different equilibrium frequencies over the alignment of our phylogenomic data compared to LG plus G model in divergence time estimation. So we conducted the simulations by verifying different factors that could affect the generation of the time tree, the length of the alignment, and the different uh, calibrations. So importantly, we focus on uh, the tree of 20 tips, and we vary the root ages from 1.0 billion years to 4.0 billion years to reflect the different timescales of the evolution. And all the sequence are simulated under the mixture model, unless otherwise uh, indicated. So we used final hazing plus MCMC tree to infer the divergence time. Here is the main result. So we compared the accuracy in divergence time estimate by verifying the root age in simulation from 1 to 4 billion years. So we have five set of calibration strategies. So let's focus on the last two, which is the root age of 3 and 4 billion years ago. So for the first calibration strategy, we have a only single root calibration, and for the second one root and an internal node half bounded internal node calibration, and for the third one node and one root plus one internal node fully calibrated, and the fourth one root fully calibrated and two internal nodes half calibrated, and the last one, three fully calibrated calibrations. And you can see that if in simulation, if the root age is over 3 billion years ago, we see that the mixture model LG plus C60 performs significantly better than their homogeneous counterparts. And in terms of calibration misspecification, we see that in case of older biased calibrations, the difference in their performance becomes even larger. But when the calibrations are set toward the present, they behave very similarly. And also, uh, what about the sequences simulated under the simple substitution models? We can see that uh, the mixture model performs equally well compared to the true substitution model, suggesting that overfitting is not a big problem in terms of the time estimate. And we also applied a mixture model and a non-mixed model on a empirical data. We found that uh, in terms of the coevolutionary timeline between the rhizobia and host legumes, one estimated using a non-mixture Lg plus G model, the divergence time, the origin time of rhizobia became slightly earlier than their legumes, which is less evolutionary possible. And this pattern disappeared if we applied a mixture substitution model. Okay. In addition, I'd also like to briefly introduce another method to approximate the hazing, which is based on the bootstrap method. This is based on the fact that under large sample size, the hazing uh, is approximately minus the inverse of the variance of the branchless estimate. So we can simply use the bootstrap estimate of the variance of the branchless to approximate the hazing. And it has a very good merit that it works as long as the log likelihood of the data can be correctly calculated using the model that you are specifying. So at the end, as a comparison, so as I mentioned earlier, so we have the very fantastic IQ2MC, which is developed by the team of IQ3. So this one is the most fast among all the different methods, and also it has implemented almost all the mixed model and also non-mixed models available in IQ3. And but again, all of them rely on the amazingly fast MCM, which is approximate likelihood dating method. Okay, so I would say IQ3, the IQ3 MC method is going to be the future thing. Uh, and I would like to thank all these people, particularly my collaborators Andrew Mead and Highway, for these two papers. And I would also like to thank Sandra, Min, Puma, Mario, Charlie, and Zheng for their encouragement and help in my projects. And particularly I would like to recommend everyone to try the amazingly fast IQ2MC if you are working on molecular clock dating. Okay, thank you very much.
Yeah, thank you, Sishu, for the wonderful talk. So, since we are out of time, we are moving to the next speaker. Thank you for a wonderful talk. So, next speaker is Pragyadeep Roy. I welcome him for his talk.
All right. Yeah. I hope my slides are visible.
Yes.
Uh, okay. Hello, everyone. Hello, everyone. I'm Pragyadeep. I'm a PhD student in Dr. Janvi Joshi's lab in CCMB, Hyderabad, India. And today I'll talk about uh, the spatial patterns of diversification rates in the Western Ghats. But I'll start off with a very well-known fact that there's immense diversity across life forms, and it is the tropics that hold a substantial amount of this diversity, right? And the process through which diversity gets generated is known as the diversification, which is basically a balance between speciation and extinction. So speciation leads to the increase in diversity, and extinction leads to the decline in diversity. And there can be different phases in the evolution of a particular group of organisms, where there can be a gradual increase in the speciation in the number of species, and there can be a sharp increase in the number of species as well. And similarly, it goes for diversity declines due to extinction. So, given a phylogenetic tree, thanks to over 3 decades of research, we have the birth death mathematical models to probabilistically estimate speciation and extinction rates. And then we can essentially understand how these rates vary across time. And there's enough literature indicating that the diversification rates, speciation and extinction, both of them can vary across different kinds of taxa and different kinds of lineages within a particular group of organisms. And then they can also vary across space. And these are global and large scale level patterns. But we can, as we move towards the regional levels patterns, the same dynamics hold true, where the speciation and extinction rates can vary across time, taxa, and space. And it becomes more interesting for regional level patterns because endemic radiations are present in every region, which are products of in situ diversification or diversification that happens exclusively to a certain landmass. And these radiations bear signatures of regional patterns and selection pressures. And neotropics are classic example of understanding the diversification patterns of endemic radiations. In this regard, one of the underexplored landmasses is the peninsular Indian landscape, which comprises myriad kind of habitats, starting from the evergreen forest to the dry savannas, and it also consists of one of the global biodiversity hotspots, which is the Western Ghats. And interestingly, India also has had a complex geological past, where it was a part of the Gondwanaland supercontinent. It drifted in isolation for over 50 million years, and then it collided with the Asian landmass finally. And because of which we have organisms of varying biotic affinities. We have Gondwanan organisms and we have Asian biota as well. So, given this setup, it intrigued us to actually address the patterns and drivers of diversification dynamics happening in peninsular India across taxa and across time and across space. So, today I'll be majorly talking about space, but I'll like to give a small glimpse of what happens across taxa and across time. But so the data uh, that I used was basically 33 dated phylogenies of well-studied radiations well-studied endemic radiations belonging to different invertebrates, herpetofauna, and zoosperms, and of varying biogeographic affinities and existing across different kinds of habitat types. So, firstly, what we did is we used 3 different birth death model frameworks to compute the overall diversification rates across each of these different radiations. And what we found out is that extinction rates were really low, and the major disparities in diversification rates were contributed by speciation. And the axis along which the diversification rates varied the most starkly is taxonomic group, where angiosperms showed highest diversification rates and invertebrates, soil invertebrates showed the lowest diversification rates. Now, coming to temporal diversification dynamics, we used these different flavors of speciation and extinction rates, where both these rates could vary in different patterns leading to the formation of various diversity accumulation profiles. Right? So what we found, so using a likelihood-based approach, we found out that out of those 33 clades, 17 phylogenies actually showed prevalence of gradual accumulation through a constant rate of speciation. So gradual accumulation in diversity uh, through a constant rate of speciation, which indicates that the habitats that these organisms have been existing in for millions of years have been very stable. But there was also good amount of prevalence of temporal dynamicity leading to the other diversity accumulation profiles, and quite a few of the lineages actually showed very abrupt episodic rate shifts, which happened at a time when the peninsular Indian landscape was going through intensified monsoon seasonality and acidification. And additionally, the dynamicity in the speciation rates was also contributed by the paleo temperature dynamics, and it became more stark towards the quaternary when there was a very sharp decline. Right? So this was all about across taxa, the patterns across taxa and across time. Now, coming to understanding the patterns across space, we specifically are going to restrict our focus to the Western Ghats for this part of the talk. So we know that the Western Ghats can be subdivided into the Northern Western Ghats, Central Western Ghats, and the Southern Western Ghats based on the vegetation type as well as the climate. And since the ancient times when there was this abrupt volcanism in the northern part of Western Ghats, till the very recent times, what has been observed is that the southern Western Ghats has been very stable by climatically, as well as it has been shielded from all these different geoclimatic changes, right? So hence we expect that because southern Western Ghats had been very stable, we would expect more number of ancient biota occurring in the southern Western Ghats, and hence it is going to behave like a museum of diversity, and hence the diversification rates are also going to be quite low, because with stability, as we found in our earlier part of the talk, that with stability, the gradual pattern of diversification is also prevalent, right? So additionally, the diversification rates could also vary across different geographical and other climatic variables like annual temperature, precipitation, etc. Right? So in order to address these questions, I actually took a subset of the taxa which I considered for the first part of the talk, which consisted of its total of 17 arthropods and chordates, summing to a total of around 590 species. And I basically collected the occurrence data for all these different species belonging to these different radiations from published public databases like GBIF, and this is basically a representation of their occurrences. So given the geographic coordinates of all the occurrences, I computed the range polygons, or basically species ranges, using dynamic alpha hulls. And from each of the phylogenetic trees, I used heterogeneous birth death models like BAM, CLADES, and MISE to calculate the speciation and extinction rates of the tips and internal nodes. And one of the examples of the range polygons has been provided on the right-hand side. And then I overlaid a spatial grid of 25 into 25 kilometers square on a region of interest, which is the Western Ghats. And then we computed the grid-wise averages for latitude, longitude, elevation, climatic variables, speciation rates. So essentially, I computed the average values across all the grids that were there in the Western Ghats landscape. And then I basically performed linear spatial mixed modeling to understand the spatial structure across latitude and longitude for the speciation and extinction rates. And then I basically tested and basically I wanted to understand what could be the relative effects of different geoclimatic factors. So coming to the results for the coordinates, temperature, seasonality, and range size was found to influence speciation rates in chordates, which showed very significant effect sizes. And also, just looking at the distribution of the speciation rates, what we found out is that the northern parts of Western Ghats seem to have higher diversification rates as it was expected. Now, coming to the results for arthropods, same temperature seasonality yet again seem to be a strong predictor for the speciation rates. However, the effect was opposite in comparison to the chordates. And additionally, what we found out is that the speciation rates actually were quite uniform across the entire landscape. However, in the northern parts, we did see some sort of for few of the regions, there were higher diversification rates. Now, as a consequence of in situ diversification, what happens is with the increase in diversity, the niches that are available that keep getting filled up, and because of which the niches get saturated, right? So essentially, with the increase in diversity, the rate of speciation or the speed of speciation actually decreases because the niches, there are no more niches to diversify, right? And also the extinction can keep increasing with the increase in the competition as the niches get saturated, right? So one of the recently developed methods to understand niche packing is yes,
Please
ma'am.
wrap up the talk very soon. It's already 9:30.
Yeah, sure. So using this method, we can actually find out the phylogenetic variance covariance, and we can also find out the range overlap of different species. And so essentially using this, so once we multiply this matrix, we would get a resulting matrix like this. And when we take the sums along the rows, we can actually find out how densely packed one species is relative to the other species in that particular radiation. So what we found out is that for arthropods, it was in the northern parts that the clade density was quite high. However, the other regions of Western Ghats didn't show much of this niche saturation, whereas for the chordates, we found out that the clade density was uniform across the entire landscape. And then the question that comes in the mind is, does this vary across time? So what I did is uh, 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 I actually performed range evolution using this newly developed method called SBS, where I could, given a phylogenetic tree and the range polygons, I can actually find out which grid cells did each of these ancestors occur with what probabilities. So essentially, I can basically get grid cell probabilities for each of my ancestors, and I specifically took the 90% credibility ranges for each of these ranges, for each of the ancestral ranges, and then I performed a time slicing approach on the phylogenetic trees, and then I computed clade density through time. Similarly, I performed time slicing on the speciation rates to find out speciation and extinction through time. And then I basically this is this is the illustration of how plate density changes with time for one of the endemic radiations that we took in a study. And in order to understand the association between speciation rates and clade density, because I had taken time slices of for every 0.5 million years, I essentially had a curve for the mean clade density through time. Similarly, I had a curve for the speciation rate through time. And what I did is, so I can consider non-overlapping windows of 5 million years, and then I can compute Pearson correlation for each of those windows, right? So what I found out is that for the chordates, the temporal association between speciation rates and clade density increase gradually towards the present. And for the chordates, what we found out is that it was the time when there was onset of monsoon seasonality and onset of intensified aridification. It was that time when there was there were many radiations which actually showed a high association between speciation rates and clade density.
Hi,
Right?
Pragyadeep. So the time is already over. So can you just quickly wrap up and finish the presentation? Because
Yeah, exactly. So this is my conclusion slide. So essentially we found significant latitudinal structure, and there were many climatic predictors which predicted the structure in the in the speciation rates. And so the clade density and speciation weights varied in different in piecewise manner and not throughout the entire history or evolutionary history of organisms. And in terms of the future work, I would be looking at the structure, how it changes through time. Yeah, thank you.
Thank you very much for the wonderful talk. Laura, please start the presentation. Yeah. So whenever you're ready, you can start the presentation.
Okay. So you can see that okay?
Yes. Yes.
Okay, so hi everyone, and yeah, so thanks. So today I'm going to be talking a bit about some work that I'm doing together with Mario. It's basically looking at an approach for fast divergence time estimation using Hamiltonian dynamics. So yeah, as we all know, in like a Bayesian divergence time estimation, we typically want to sample posterior distribution of our like node ages, our clock rates, and our branch lengths. But you know, this posterior is analytically intractable, so we have to use these expensive MCMC algorithms to sample from it. So what I've been looking at is basically like how do we efficiently explore this high dimensional parameter space? So basically during our MCMC, what is the best way to sort of sample through this parameter space? So yeah, one of the most common approaches that's used is this Metropolis Hastings sampler. And this kind of works by you essentially are exploring the parameter space in essentially like a random walk, and you're updating your parameter values like one at a time. So if we do like an example of an MCMC, I'm sure we're all quite familiar with this image from Paul Lewis's website. Basically, you know, you're sampling, suggesting different values, and the blue ones are like accepted, and the pinky ones are like rejected samples. And yeah, essentially, you know, you're going to get to areas of higher probability, but we know we have a lot of like rejection sampling, and all these pink ones basically aren't being, are being rejected. So while this is fine for kind of like smaller data sets, as you kind of have larger data sets available now, with like higher number of dimensions, Metropolis Hastings becomes quite inefficient as it's like rejecting lots and lots of samples. So it's like it's not exploring it very well. So a lot of things are getting rejected. And this also just wastes like a lot of computational time because yeah, things are being rejected, but we still need to calculate like the likelihood, the prior, and everything just to ultimately reject it. So like for Metropolis Hastings, we're often seeing like, you know, maybe like 20 to 40% of samples being accepted, which also means that like 80 to 60% of our computational time, is not being accepted. So yeah, this is okay for like smaller data sets, but yeah, data sets are getting a lot bigger. So kind of our ability to like sequence genomes and the effort in genome sequencing is growing rapidly at the minute. So we have like, yeah, you know, like the Bird Project, Human Genome Projects, all these different things producing all these like genome level data sets. And essentially now like our methods are struggling to keep up with the amount of data that we have, and they're kind of becoming the bottleneck. So yeah, we need ways to kind of increase how we can sample. So like, for example, for this analysis here, they actually ended up like kind of doing like computational tricks, so like sequential sampling. So, you know, kind of like splitting up the tree into different kind of subsets, doing your inference and then kind of patching it back together. So while this works, it'd be nicer to have one kind of like unified analysis that we could do our divergence time estimation in. So for that, we've been looking at using a Hamiltonian Monte Carlo sampler. And this HMC sampler is basically an idea is borrowed from physics. And you can think of like a particle kind of like rolling around on the surface of your posterior distribution, and allows us to, instead of making these like random jumps, we make kind of like guided steps through the posterior. So it's essentially like, kind of think of it as like we give our particle like a random momentum, and then we run it along the flipped posterior surface. So then the particle kind of naturally gets stuck in areas of high probability. And it's a way of like avoiding these kind of like poor samples, poor proposals. I'll explain it in a bit more detail in a few slides, but essentially, this was the concept that we wanted to use. So we developed HMC div, which is like an R package with C++ backend, and it's a hybrid MCMC sampler that allows us to sample our node ages, our branch lengths, and our rate model parameters. So it's on a fixed tree, basically doing divergence time estimation. But we use these two different sampling strategies. So for node ages and the rate and like the clock model parameters, we use a traditional Metropolis Hastings. But for branch lengths, we use the Hamiltonian Monte Carlo sampler for the more efficient sampling. And this also allows us to update all parameters in each step. So just to compare kind of like HMC div with MCMC tree, which I'll do a lot during this talk because it's kind of like the fastest approach for divergence times at the minute. So we have two moves on our node ages. So a mirror move and a mixing move. And these are implemented the same way in both software, this like stepwise proposal for the mirror move. And then we have like a global proposal for our mixing move, which moves all the node ages at once. And for our clock parameters, we have the stepwise. And then it's for our branch lengths that are really different. So in HMC div, we jointly estimate all of our branch lengths in one step, and in MCMC tree, it's a stepwise proposal. So this is the part that like really, really speeds up HMC div relative to MCMC tree. So basically, for branch lengths, um, with HMC div, you have one likelihood calculation and then 2 to 6 gradient calculations, which are quite cheap, as opposed to MCMC tree, which are going to have like almost 2,000 likelihood calculations, like to sample every branch. So yeah, you can see it's an awful lot more like computationally heavy in MCMC tree than in HMC div. So just to explain a bit more how it actually works. So as we heard about in the first talk about like the Hessian and everything, so we still need to estimate our Hessian as we would for like doing an approximate likelihood. So yeah, we can use IQ tree or CodeML or BaseML. Then we can input that into HMC div. And then so this plot here basically shows the likelihood surface for two kind of branch lengths. And the contour lines here are based off the Hessians that's estimated. So you can have like a tight Hessian, which is like the one in green, or like this flat one, which is in blue. So the center here is basically the maximum likelihood estimate for your branch length. And then, yeah, the shape of the contour line is controlled by the Hessian. And it will give information on how controlled kind of like how much information the data gives about that branch length. So to make a bit more sense, kind of if you look at it from this way, this is in HMC div, you kind of work in this like potential energy space. And yeah, say this is like these Hessians here. So for the steep one, this would be the steeper one that controls like the shape of this bowl. And this is the shallower one. So if you kind of imagine that like your maximum or like your highest probability is kind of down at the base, and then yeah, so you estimate basically the branch lengths along here. So like in practice, basically, yeah, you start kind of at one point, and then you use like a leapfrog approach, which says, okay, you get some momentum, you start your sampling, you do like a half step, and then you calculate the gradient. So the gradient will tell you where along this curve you are, right? So if you're here, it's going to kind of push you down. If you're here, it's going to push you down here. It's kind of keep you kind of in these areas of higher probability. So you kind of do a half step, calculate the gradient, a full step, calculate the gradient, and a half step again, calculate the gradient. And in this way, you kind of sample like quite largely across the posterior distribution, but you're kind of kept in the areas of higher probability. And this is just showing for like two branch lengths, but of course, if you're doing an actual analysis, this will be a very high dimensional space, and you'll have them for all branch lengths. So yeah, basically, you move from one step to the next, and then the move is accepted based on this equation here. So basically says the potential energy plus kinetic from where you started minus from where you ended up. And in theory, this would be completely conserved. You conserve all this energy, so every move would be accepted. But because we're using an approximation, there is some rejection that's going on, but it's still much more efficient. So you still see like 80 to 90% of your steps being accepted. So yeah, the advantages of this then become twofold in that statistically we have fewer rejections because we have these kind of like gradient kind of guided steps through the posterior. We're often accepting our steps, and also it's computationally faster because we've so much fewer calculations. So yeah, so what I set out to do was basically benchmark this HMC div approach with MCMC tree, and we used the approximate likelihood MCMC tree because that was kind of like the fastest approach. So we have two fossil calibration scenarios. I looked at basically a single fossil calibration on the root, and then multiple fossil calibrations along the trees. And then we evaluated based on the correctness, so ensuring the two software converged on the same values, and then the efficiency, so like compare the ESS values per iteration. So these are the data sets that I used. So basically 4 genomic data sets, they're different size, different partitions, and different number of taxa. So yeah, we have 3 mammal groups and 1 birds. So basically for the one calibration, I just set like bounded kind of root calibration. But then for the multiple calibrations, I used actual calibrations from the papers I took them from. And these are all the results I'm going to show are just using the results with multiple calibrations. So when I looked at the correctness, yeah, basically ran both HMC-Div and MCMC-Tree until they reached convergence, just to ensure that both programs converged on the same parameter estimates. And this is just, I'm just going to show one of the results here. All the different data sets show the same pattern. So basically this is the node ages plotted against each other for those estimated for HMC-Div and MCMC-Tree. So this is the posterior means, and then we looked at the 95 and 5% quantiles. So this is just to show that, yeah, using the two programs, we are getting the same values estimated for our node ages, and similarly we get the same for the rates. So yeah, this was good. So then we wanted to look at the efficiency. So we basically ran both HMC-Div and MCMC-Tree for 100,000 iterations, and we sampled every iteration. And this allowed us to compare basically the rate at which each method accumulates effective sample sizes. So this is kind of one of the results, again, for the multiple calibrations. And this is looking at the efficiency, so the ESS number per iteration. So you can see, oh yeah, also, so like the dash lines are both software without the mixing move. I don't really have time to go into the differences between the moves, but yeah, basically, if you look at this line here, this is HMC-Div and MCMC-Tree. So you can see the efficiency of HMC-Div is a lot higher relative to MCMC-Tree per iteration. And yeah, I do notice that we see this drop here. But yeah, these are like empirical data sets that have different calibrations, different setups. So like you wouldn't really expect that they'd be definitely linear. I've done some like simulations to show kind of a nicer pattern, but yeah, and then we can also use this information to basically understand how long it would take if you want to say, okay, if you wanted each program to get to have 1,000 ESS values, we can calculate that. So yeah, you can basically see becomes where HMC-Div becomes much, much faster. That basically you can have, yeah, so if you look at this data set, it takes 0.007 days using HMC-Div versus 29 days for MCMC-Tree. So it's almost like 4,000 times faster using HMC-Div relative to MCMC-Tree. And similarly with this one, it's like, yeah, 1,500 times faster. So it's really, really having like quite a big speed up relative to MCMC-Tree. So yeah, basically we've implemented this R package. We're still working a little bit on kind of having like empirical case studies. Yeah, we're just trying to get it all set up. So, but it is allowing us to estimate divergence times of these large genomic data set under the single unified inference. And yeah, a nice thing, of course, it's saving computational time, so reducing the amount of computational hours that we need to log up. And yeah, with that, I just want to say thank you to yeah, especially Mario, Sandra, and Zihang, who've helped a lot, and yeah, everyone there. Thank you.
Laura. Thank you for sticking to the time. I invite Hannah Dugdale for the next presentation.
I want to start the presentation.
Yeah, please start.
Yeah, I don't seem to be able to share my video, but okay. Hi, I'm Hannah Dugdale. I'm at the University of Groningen, and it's a great pleasure today to present this work on the survival costs of mothers of having sons in pre-industrial Finland. So this is a really nice, proud supervisor moment because this work was done by two amazing students. Ewan Young was my PhD student. He's now finished his PhD. And Leon van Dorp was a master's student, both at the University of Groningen. And it was a collaboration with Mirke Ladempere and Vepi Lamar, who are at the University of Turkey. And if you want to know more about it, there's a QR code here to the preprint, which is available on bioRxiv. So life history theory tells us that individuals will face fitness constraints when they invest in reproduction. So this means that when individuals have a limited amount of resources and they invest in reproduction, they have less energy to invest in their own self-maintenance and survival and future reproduction. So a variant of this is the expensive sons hypothesis. And this says that mothers will have greater fitness costs when they produce sons instead of daughters, when it occurs in a species with male bias size dimorphism. So this means that when males are larger and more costly to produce than females, mothers should show a reduced survival cost of producing these sons. So what is this true in humans? Is there a higher cost having sons over daughters? Well, it's clear that this is true. So sons cost about 10% more energy for mothers during pregnancy and about 7% during lactation, so about 5 grams extra breast milk per day. But if we look in the literature, there's very mixed evidence for the expensive sons hypothesis. So if we just look in humans, there was a nice review of this in 2024, and this listed 17 studies, and only 5 of those studies showed a higher cost of sons over daughters. So why is this? Well, there are several previous limitations, mainly that prior studies have looked at the long-term fitness costs. So they've looked at the probability of producing sons, changing the postmenopausal lifespan of the mothers. So it's a very long-term survival fitness cost. And the shorter-term costs have been limited to reproductive factors, such as the effect of sons on future reproduction. So the survival studies that have looked at mothers that reach menopausal age have been looking at the cost after menopause. But there's actually no real reason that these costs should only occur after menopause. And in fact, there's evidence that these should occur immediately after birth. So, for example, the occurrence of sons increases the likelihood of cesarean, which is a life-threatening condition to the mother and offspring. So what this means is that if we're focusing on menopausal women, it means we're going to be underestimating the costs of sons because we're not looking at the costs during reproductive years. And it might bias the data set that we're looking at and bias estimates that result from our analysis. So, for example, if we're excluding frail mothers that die before reaching menopause. So we had one main hypothesis that maternal survival was going to differ for mothers. And by maternal survival, we mean survival within 1 year of last birth, so looking at short-term survival. And we thought that this would differ for mothers whose last birth was a son versus a daughter, or differ according to the number of sons and daughters that a mother produced over a lifetime. And we also hypothesized that this would be moderated by socioeconomic status and the number of children that a mother has. So the idea here is that mothers who have a son at their last birth would have reduced survival, that a higher proportion of sons would have reduced survival, and that this would be exacerbated by mothers of lower socioeconomic status and those who had more children. So we use this amazing data set which Verpi Lamar has, which is of parish level birth, marriages and death records from rural pre-industrial Finland. And this spans 23 different regions in Finland that you can see on the map. So we had data from 5,546 mothers, and they were mothers who had a last birth from 1749 to 1889, which are quite precise dates. But in 1749, the King of Sweden, who ruled at that time, wanted to be able to tax people. So it was required by law for the Lutheran Church to record these birth, marriages and deaths. And in 1890, the industrial period started in Finland. So we use the pre-industrial rural data where there's high fecundity and mortality, and the median number of children for mothers in our data set was 5, but the maximum was 17. And the median lifespan from birth was 24 years. But this is higher for mothers because a lot of this mortality was from children who died before their 15th birthday. So 40% of children never reached their 15th birthday. And this was primarily due to infectious diseases. So, for example, the smallpox vaccine didn't come around until 1880. So if we look at our data, we can see that 8% of the mothers died within the first year of their last birth. And half of these mothers died within 42 days of their birth. So it's a very short time frame, primarily from the birth to death. And of these mothers that died after the last birth within a year, 52% of these last births were to sons. So we use this data set to test whether maternal survival was reduced in mothers whose last birth was a son, who had more sons than daughters, and whether this effect was moderated by socioeconomic status and the total number of children. So our first finding was that we did not find an effect of the last birth being a son. So maternal survival was similar for mothers who had a son or a daughter at last birth, but there was effect on the proportion of sons that they had in relation to the number of children. And these analyses all accounted for the age of last birth of the mother, last birth year, the geographical region in Finland, family ID to account for sisters in the data set, and whether the birth was a singleton or multiple birth. So this is the main result. What you can see here is that mothers who had just one offspring had a similar survival rate, irrespective of whether they had a son or a daughter. But as we go into larger family sizes, families of 4 or the medium of 5, there was an expensive son cost. So mothers who had more sons, that's the yellow line here of 1, had lower survival than mothers who had just daughters. And this effect actually started to reduce and then actually turn around in larger family sizes. But you can see the error is massive because we've got few sample sizes here. So just to zoom in on this graph, just to show you a bit more clearly, here is the median number of children, family size of 5, and mothers who have more sons have got a reduced survival 1 year after birth than mothers who just have daughters. And we also analysed this data set excluding family sizes of more than 10, and this relationship still held. So the other moderator we tested was the socioeconomic status. So this didn't have an effect on the cost of sons, but there was a relationship with the cost of children in that mothers of intermediate social status had reduced survival when they had larger family sizes. But again, this is in larger family sizes were quite rare, and if we analyse it just up to the 95th percentile, we don't see this relationship. So overall, what we found is that there is evidence in our data set for the expensive son hypothesis, but it's moderated by the number of children that a mother has, and is in relation to the total number of sons and daughters a mother has. So we see this reduced survival in mothers with a median number of children and that had more sons than daughters. And this effect size actually weakens and reverses at larger family sizes, which is not what we predicted. But it may be suggested in our data set we have selective disappearance of frailer mothers who don't reach these larger family sizes. And this finding is also consistent with a recent study in Canadian mothers, which showed that mothers who had poorer health, where there was higher infant mortality, showed greater costs of having son, which suggests we're looking at biased data sets if we're looking at larger family sizes and those individuals that reach menopausal age. So the implications are that studies that just focus on mothers that survive to menopause are actually going to underestimate fitness costs of having sons, or fitness costs in general if we're looking at reproductive costs, because we're not looking at the cost during reproductive years. And if you die during reproductive years, that has a massive cost on your reproduction. And it may actually result in biased estimates if we're looking on mothers that reach menopausal age, because we're excluding those frailer mothers who died during reproductive years. Overall, the costs of survival were actually quite small. So it was about 0.4% reduction in maternal survival within a year of birth per son for mothers that had a medium family size. But this effect was actually detectable and actually is likely to be an evolutionary constraint on family size. So that was it. Thanks for listening to the talk. And if you want to know more, please have a look at the preprint.